New technology keeps some of these insects, which spread malaria, from reproducing. A new genetic tool may help wipe out Africa's main malaria-carrying mosquitoes. It's known as a gene drive. In tests, it sterilized Anopheles gambiae mosquitoes. In other words, it kept them from reproducing. A gene is the basic package of genetic instructions that tells cells how to build some protein. Proteins are the chemicals that do much of the work in cells. Gene drives are pieces of DNA that have been tweaked by scientists. These snippets of DNA have been designed to find a target gene, slice into it, and then insert themselves. This cut and paste system alters the function of the original gene, and it is self-propagating, which means that it makes more of itself. Other human tweaked or engineered genes don't reproduce themselves. A gene drive's ability to make more of itself helps it get into more mosquitoes than would a regular inserted gene. This is the second gene drive aimed at knocking out malaria. The first, announced 2 weeks earlier, works quite differently. It stops mosquitoes from transmitting the parasite that causes malaria. The newer gene drive instead eliminates the mosquitoes themselves by making it impossible for females to make new baby mosquitoes. Scientists reported their achievement December 7th in the journal Nature Biotechnology. Technique appears potent. Most genes come in 2 copies. One copy comes from the mother, the other comes from the father. So each of the parent's genes has only a 50-50 chance of getting passed on to a kid. But like Star Trek's Borg, gene drives become part of every unaltered target gene they encounter. These ambitious bits of DNA break standard inheritance rules. With the new technique, 76.1% to 99.9% of the offspring inherited the drive. As a result, the edited genes drive themselves quickly through populations. Austin Burt first conceived of the idea for gene drives in 2003. This evolutionary geneticist works at Imperial College London in England. For more than a decade, gene drives remained mostly just an idea. But this year, thanks to precision molecular scissors, known as CRISPR-Cas9, 4 gene drives were put to use in several labs. These included the 2 in mosquitoes. They all work terrifically, says George Church. He's a geneticist at Harvard University in Boston, Mass. Cas9 is a DNA-cutting enzyme borrowed from bacteria. Researchers can design genetic cousins of DNA, molecules known as RNA, to guide the enzyme to desired genes. Not ready for prime time. Church is pleased to see that this latest mosquito gene drive works. Still, he says, further tweaks may be needed before it is ready for release into mosquitoes in the wild. Researchers also may want to combine approaches. They might first release a gene drive that would prevent mosquitoes from carrying malaria. Later, they might release another to control mosquito reproduction, he suggests. In the new study, Burt and his colleagues first used CRISPR-Cas9 and another type of gene editor known as talons. These disrupted each of 3 genes that are very active in the egg-making organ of mosquitoes. Females carrying 2 copies of any one of the 3 disrupted genes were sterile. Once the researchers had confirmed that messing with these genes kept the mosquitoes from reproducing, the team then built gene drives to insert them into the genes. Gene drives that interfere with reproduction make some people nervous. Such drives could make a species go extinct. Many people would not miss mosquitoes, but no one really knows what getting rid of the insects might do to their ecosystem, the animals, plants, and other living organisms in their neighborhood. For instance, some of those neighbors might rely on mosquitoes as lunch. This gene drive also has some technical glitches. That means it won't be the final version used to control wild mosquitoes. But the scientists are hopeful that future gene drives could cut populations of malaria mosquitoes dramatically, says study co-author Tony Nolan. He is a molecular biologist also at Imperial College London. The public health community needs new approaches for mosquito control, he says, and this is a promising one.